So forgive the possibly unimaginative title, The Good, the Bad, and the Ugly, but I did think that I could mention some of that. Botulism isn't all bad. There are some good sides to it. Well, certainly to the botulinum toxin. But I also thought of calling my talk, now it doesn't want to move. Yeah. What I know about botulism, because when I was asked to give a talk on botulism, I could think of about 3 things I knew, and that was it. So it's been quite exciting learning about it. All I actually previously knew about botulism was it had something to do with honey and not giving honey to babies under a year. I preach that all the time to students and say, apparently you mustn't give babies under a year honey, but I knew it had to do with botulism. I wasn't sure exactly why. I knew about blown or bulging tinned food and that people worry about botulism. And I obviously have also heard of Botox for cosmetic and also therapeutic purposes, but I didn't know much else. So I'm just hoping, other than the experts out there, that there's a few people who know as little as I knew, and that I can tell you something interesting and new. So today I'll be covering the history, just a little bit of the history of the discovery of botulism. I'm going to talk about the organism, the toxin it produces, the pathogenesis, epidemiology, the different disease types and how it presents, then the diagnosis, management, and the prevention. So the history, just a little bit of the history, is that in Latin, the word botulus means sausage, and that actually is a sausage-shaped organism, although it possibly, you could say it is. But it's actually because there were outbreaks of the illness in the 18th and 19th century that were noted to be related to eating sausages. And this man here, Justinus Kerner, was a German district health officer and also a poet. And he noted the connection between a paralytic illness that occurred in 230 patients in 1820 and the fact that they'd all eaten the same sausage at a meal. And so initially he called it sausage poisoning. And later, another German physician coined the term botulism. Russia, a similar illness was linked to fish poisoning. Then in 1897, a Belgian bacteriologist van Erminghem described Clostridium botulinum that he'd isolated from contaminated ham that had poisoned people. And he also showed that it produced a toxin which caused weakness in animals. There were a few events along the way, but I thought it might be significant that they then isolated the type A botulinum toxin and purified it in 1946. And then in 1980, Dr. Alan B. Scott used the toxin for the first time in humans to treat strabismus. Okay, so the organism is a large gram-positive bacillus, which means it's rod-shaped. It's strictly anaerobic, which essentially means oxygen is toxic to the organism, but because it's quite a devious organism, it can generate a small amount of oxygen due to the fact that it has superoxide mutase, and therefore it can bypass some of the cellular mechanisms where our neutrophils produce oxygen radicals. It's also motile, and it forms subterminal spores, which is important, I guess, when you're looking under a microscope and you're seeing gram-positive bacilli, because different gram-positive bacilli have different appearances of where their spores lie. So subterminal implies it's not at the very tip of the organism. And the spores are a very important feature of Clostridium botulinum. It goes into the sporulation phase, and then when it comes out of it, it's called the germination phase, when it's in a vegetative state, and that actually means it's a growing, reproducing cell. They form spores under certain conditions. When they decide things aren't optimal, they use fancy intercellular communication, quorum sensing, and all the organisms activate or repress various genes which initiate spore formation, and when the time is right, then they germinate again and become more active and dividing. And the toxin is obviously produced by the growing cells, not when they're in their spore state. So the spores are key to the organism's survival, and because of this, they can exist for millions of years in extreme conditions, resistant to heat, cold, desiccation, exposure to light, and they're ubiquitous. They are found in soil all over the world, marine sediments, water, and because it's in soil, it's likely on all fruit and vegetable and food surfaces, and in the intestines of animals. So it's all over. And it's not actually killed by boiling at sea level or 1 atmosphere for even several hours. In fact, that can even make the environment a little bit more anaerobic and actually favor their growth. But active growth is suppressed by pH below 4.6, a high salt content of the surrounding fluid but it must be at least 10%. Increased sugar environment and a temperature below about 3 degrees Celsius. This won't kill the organism, it just into that spore state, the sporulation. And then germination occurs when it switches to the vegetative phase. And this happens when there's an absence of oxygen, because the organism likes this. It likes a temperature somewhere between 15 and 37 degrees Celsius, and it likes a less acidic pH, around 7. So this is just a diagram showing the different phases, the sporulation and then the germination. So there's your growing active cells, and they're the ones that produce the toxin. And when they decide they want to make a spore, it's the DNA replicates. So it makes 2 cells, essentially, one larger than the other, which is called the mother cell, and that's going to become the free spore. And the mother cell then engulfs the free spore. And then these little vesicles, which contain dipicolinic acid, which is very important for spore formation, they get incorporated into it, make this nice, sturdy, hardy coat. And eventually this spore breaks free from the mother cell, and it can become this dormant, survive in whatever conditions until the time is right. It becomes rehydrated and becomes a vegetative cell again. Other genetic things happen as well. Things are switched on. Okay, so the Clostridium, one organism. It's actually a species which is divided into 4 genotypically and phenotypically distinct groups, but there's a whole lot of different organisms. What's common to them is obviously that they're all gram-positive spore-forming bacilli, and they produce the botulinum neurotoxin. There's also a Clostridium butyricum and Baratii, which also produce a toxin. And there are 8 toxins produced by these various organisms. It's A to G, and there's a C1 and C2, so that's why there's 8. And within those, there's A, 1, 2, 3, 4, and various numbers of toxins. I hope everyone can hear me, despite the rain. So the group 1 produce toxins E and F, and these are the 3 most potent toxins, being the most, and then in descending order B. And lytic and culture. The group 2 produces toxins B, E, and F, and they're non-proteolytic. And these are the 2 groups that affect. Then group 3 produce toxin C and D, and they're only found in animals. And then group 4 has been shown to produce this toxin G, but it hasn't been associated with any human disease. Okay, they found that the group 1 organisms are found more in temperate climates, and they're of terrestrial origin, which is more in soil and dust. And they thrive in warmer temperatures around 30 to 37 degrees. And then group 2 are found in the northern hemisphere in aquatic environments. They have an growth at a little bit of a cooler temperature. It starts as a polypeptide chain, which actually has low potency, but then the toxin is cleaved by bacterial proteases, becoming a molecule which has 2 chains that are connected by a disulfide bond. And they vary in molecular weights, the different types of toxins, but by weight, type A is the most potent toxin found in nature because it's for its, and it is the most toxin. them in a lab. It has helped provide insights into the mechanism of how neurotransmitters are released. The botulinum toxin is very similar in structure to Clostridium tetani toxin, but they have very different effects. Botulinum toxin mainly acts on peripheral and the autonomic nervous system, and the main feature is weakness, whereas tetanus toxin works on the cells in the inhibitory cells in the CNS, and that results in rigidity and spasms. So I'll show you in the next but the head in a light chain. The chain binds to the presynaptic receptor. Just to know, toxins act at different receptors. It's endocytosis is cell drinking. It's sort of drunk. It's taken into the cell, and what normally happens is an action potential will trigger influx of calcium, of synaptic vesicle contents, which is the neurotransmitter. And what clostridial toxins do is they cleave the crucial peptide bonds, which allow release of the neurotransmitter and thereby prevent the release of it, which in this case is acetylcholine. So stimulation of a presynaptic cell, which is usually the alpha motor neuron, produces no neurotransmitter release. So nothing happens, and there's paralysis, autonomic dysfunction in parasympathetic nerve endings, and also in autonomic ganglia. So in this picture, this is the terminal of the axon terminal with all these presynaptic vesicles containing acetylcholine. And this is the synaptic cleft, and this is where botulinum toxin ends up. Normally, what you need is a group of proteins which will bring this vesicle to the membrane, attach it, dock it, actually and connect it, and then allow it to spill out its contents into the synaptic cleft. But when botulinum toxin comes along, it binds to the membrane at a receptor, it's taken in, and then the light chain cleaves these proteins, and then they can't allow any connection of that neurotransmitter vesicle, and there's no release of neurotransmitter. Originally, it was thought that once this happened, the synapse would be rendered useless, but they've done a lot of research into the mechanisms of recovery, especially since botulinum toxin is so widely used in medicine. So they've seen that there's actually sprouting of the presynaptic axon with the formation of a new synapse. And once the original terminal recovers, these new sprouts are pruned away and they can get back to functioning. Different toxins are associated with different recovery times. The toxin can gain access to the CNS, but symptomatic involvement is rare. So it's not actually immediate, the effect, because you need, if you ingest the toxin, it actually still has to get all the way to that synaptic cleft and produce its effect. So it actually takes about 24 to 72 hours to take effect. It can be as short as 4 hours and as long as 8 days. And therefore also, I think in an outbreak, not everyone presents at necessarily the exact same time. The peak effect is at 12 days and the effects last 8 to 12 weeks. I keep wondering, is that if there's treatment, if there's not treatment, but I think these figures are based on the assumption that the patient is supported through it. And then this would be the natural course while you're supporting the patient. So they talk about the different kinds of botulism. The presentation, the illness is always the same, the clinical picture, but how you get it is different. So there's food-borne, there's infant botulism, wound botulism, botulism of undetermined etiology, or adult intestinal botulism. And that's when there's colonization by spores in the GIT. There's iatrogenic, and then there's inhalational. There's a potential for that because it's a good option if someone wants to, for bioterrorism. So it can be released and as an aerosol and inhaled. So the epidemiology, it depends which form you're actually talking about. Foodborne usually occurs in outbreaks, and from what I gather, when we talk about botulism, that's any more than one case. 2 or more cases in a given time is technically an outbreak of botulism. But you might not all, like I said, you won't always see all the patients clustered. If it was in one batch of food, for example, it might spread far and wide, and you might pick up cases over a period of time and over a different area, but it can also occur in a cluster. Then other forms are more sporadic. Commercially canned foods used to be the most common source, but now home canning fruit, fish, and vegetables is more commonly implicated. And it just depends on various cultures around the world, but whatever food is locally available, the preservation or the different processes can lead to botulism. So in Alaska, there's fish fermentation, and that can cause botulism. In China, homemade fermented beans. Peyote in Mexico and South America, it's a small spineless cactus which is actually eaten during various religious rites because it's a hallucinogen, and they're obviously growing in the ground. It can be covered in spores and then just ingested. And then Pruno, this was new to me, is something, it's a prison wine, and it seems to be a well-known thing in prisons, and the prisoners can take various fruits that are available, any available sugar, a plastic bag, this is a picture of it, hot water, and they basically ferment that. And they need a sock or a towel or something to drain out the pulp, and then that can get to about a level of 2 to 4% alcohol. And so outbreaks have been seen in prisons at various times. Those were in California, and then in 2012 in other parts of America. And that seemed particularly when they used potatoes. So that made it worse. But basically spores of Clostridium botulinum are ubiquitous. So it can happen anywhere. But the statistics I got were the US statistics that just to show that although we don't hear of it happening much here, it's being recorded and it's happening. There were 263 cases between 1990 and 2000, and then in 2015, 39 cases were recorded, almost always in little outbreaks. And then infant botulism, over a period of 14 years, there were quite a lot of cases, 2,419. I couldn't get numbers for these, but wound botulism will occur where there's the particular drug is used that's associated with it. And in various places, quite a few sources I saw, it said that most cases seem to be in California, but there have been cases reported from Germany. Then iatrogenic is uncommon, but it's associated with the unlicensed use of cosmetic botulinum toxin A. And then inhalational can be associated with use of contaminated cocaine that's snorted. And then it's also a potential route of bioterrorist attack, but I didn't read of any actual incidents. So what about here in South Africa? I found an article in the International Journal of Infectious Diseases from February 2021 by M.N. Vosloo, which reported the first laboratory-supported case of infant botulism on the African continent. And interestingly, it was actually a bivalent botulinum strain, which produced two toxins, B3 and F8. I looked on the NICD website, which said that the true human disease burden and incidence is actually unknown. It's said to be rare, but a number of cases have been reported. It also says many cases go unnoticed. Now, I'm not sure if that means unnoticed as in maybe mild and the patient recovers. I wonder if some patients die before they get to hospital, and also how many we sometimes see and never know or don't think of the diagnosis. If patients come in requiring ventilation, we don't actually figure it out. Maybe they demise and we haven't considered it. But it is known that it causes disease in farm animals and wild water birds, and this is locally called Lumsikte. And then there was an interesting case, which was reported by Professor John Freen in 2004, of a fatal type A botulism in Springs, South Africa, that had occurred in late February 2002. Where two siblings, 8 and 12 years old, both developed acute flaccid paralysis and died. One died, I think, almost immediately, and then the other one survived for about 10 days on a ventilator, but demised also, unfortunately. And the toxin was isolated from the food, which in this case was found to be a tinned fish in tomato sauce. And they also cultured C. botulinum toxin, cultured C. botulinum organism from the food. So they found the toxin and the organism, which is the important thing. And it was actually investigated and found the corroded and perforated area on the tin, which is likely where contamination occurred. So the first outbreak This was said to be the first outbreak of human type A botulism in South Africa that was documented, and the first fatal outbreak described. There had been previous botulism cases in humans described in the region, but they had been milder. And there have been a few other outbreaks in Africa reported, namely a type E epidemic in Egypt. So the disease, the classic presentation of the disease, I think it's pretty specific. If they have all these features, it's usually acute, if not, then it's subacute, bilateral, symmetrical cranial neuropathies, which are associated with symmetrical descending weakness. And there's a few things that seem to be important, that there should be an absence of fever, there should be symmetrical neurological findings. The patient is still responsive, although this may be difficult to see because their face is expressionless. So they might seem unresponsive, but their CNS is actually uninvolved. The heart rate is normal or slow in the absence of hypotension. So they won't have a tachycardia. And sensory deficits are absent, except for the description of blurred vision, which isn't, it might seem like that's sensory, but it's actually a motor deficit. Occasionally, because things don't always follow the rules, you might find asymmetrical findings and ascending weakness in 7%, up to 7%. It typically starts with nausea, a dry mouth, and there may or may not be diarrhea. The diarrhea is not from the toxin. The toxin produces constipation. But because this is spoiled food, there might be other acids or organic products that have been produced, and that is the cause of the diarrhea if it's present. The eyes, I'm not quite sure how these patients actually complain of this if they are so weak, but maybe before it gets too bad, they complain of blurred vision. They have dilated pupils in up to 50%, and they say that can actually last for months after recovery even. And the reason it's blurred is they have lack of accommodation, and they also have ophthalmoplegia, so cranial nerves 3, 4, and 6 involvement, ptosis, and there might also be nystagmus. So in this picture, you can see they're having to, you might not be able to appreciate the ophthalmoplegia because of the ptosis, but this patient's unable to move his eyes. They can also have obviously bulbar involvement, so dysphagia with drooling, dysarthria, and hypoglossal weakness, and then it progresses to the upper limbs, trunks, and then the legs. And then there may be respiratory compromise, and this could be from upper airway obstruction because of, flaccid weakness of the soft tissues in the upper airways, and then with or without as well diaphragmatic weakness. So the type A, which is the most severe, might even need 2 months on a mechanical ventilatory support before weaning off, and type B up to a month, but the hospital stay can be up to 3 months for these patients. There's also often autonomic dysfunction, which involves GIT symptoms, changes in heart rate, hypothermia, which I thought was interesting because I thought it's similar to anticholinergics, which can cause hypothermia because it stops the sweating. But I would imagine if they are exposed to cold and they're unable to shiver because of the paralysis, they might lose a lot of heat. And then urinary retention as well. For infant botulism, it begins a little differently, not as acutely, I think, and they can present with constipation, poor feeding, floppiness, drooling, a weak cry, and noisy breathing, stertor. And this, in severe cases, up to 50% can actually develop respiratory failure from diaphragmatic weakness. And the usual course seems to be that it progresses for 1 to 2 weeks, then stabilizes over 2 to 3 weeks, and then the recovery is thereafter. Again, like I say, I think that would be with support. This is the progression and then the improvement. Obviously, if they are unsupported, they don't receive medical treatment, they'll progress and can become paralyzed and obviously demise. In wound botulism, there isn't the GIT prodrome, but the rest of the paralysis is similar. They shouldn't have fever, but if they do, it would probably be from the wound infection from the other organisms. And the wound might not actually look as bad as the neurological symptoms, because botulism doesn't actually cause the wound, which makes me think of Clostridium tetani as well, where we see sometimes dung is put onto the umbilicus of a newborn, and it doesn't look particularly infected at all, and yet the spores have entered the blood. Well, the toxin has entered the bloodstream, but it can also produce abscesses. So the pathogenesis of the different disease types in the food-borne type, the toxin is preformed in the food and then ingested with the food. It's absorbed in the proximal GIT and then ends up in the peripheral cholinergic synapses and the neuromuscular junctions. The clinical picture depends on the toxin type, not the kind of botulism. And as I said, it always seems like a similar picture, but some are more severe than others. In the infant or adult unknown etiology type, the spores are what is ingested, and they colonize the GIT of at-risk individuals. And these at-risk individuals are infants under 1 year because they seem to have the right GIT environment, milieu, and a higher pH, which allows the germination of spores. So it seems to say after a year, when pretty much all babies above a year of age are now on a more mature or adult type of diet, the pH drops. But in that first year, it might be higher. And whatever the reason, the spores want to germinate in an infant's GIT. And then the adults who can get it would be ones who might have achlorhydria, anatomical abnormalities, or ones who've got an altered microbiota from antibiotic use. And then the toxin is produced within that patient's GIT. And in the wound type, spores will, in the wound will germinate and produce the toxin. So this was one of the uglier sides, the wound botulism. It's seen mainly in IV drug abusers, and specifically and quite particularly with the use of something called black tar heroin, which seems to be a cheaper, unrefined or unpure form of heroin, where it's actually it's a black sticky substance that's injected. And I don't know why, but it's often bulked up, so it looks like more with dirt, shoe polish, or wood pulp, which will obviously possibly contain spores. And the spores can also be contaminating drug paraphernalia. So the wound happens at the site of injection, and it often happens in drug users who don't inject into the vein. They actually inject into the tissue or the muscle. It's actually got a term, skin popping, where it's intentional because it's easier than finding venous access, and there's a slower release or absorption of the drug. And so it sits there under the skin in the tissue. Shared needles are also a risk. So obviously there's a lot of organisms present in these wounds, and it creates a nice anaerobic environment for Clostridium. And especially if there's abscesses where there's low oxygen tension, it's a good place for them to germinate and produce toxin. But there's actually only 50% have visible abscesses, but you will see the injection sites on a drug user. So this is rather dramatic wound. This is post-debridement, but it still shows a lot of tissue loss. And then this would be the clinical picture of someone who's got a lot of skin ulcers and from the injection sites, and presenting with the classic features with the ophthalmoplegia and the ptosis and the weakness. And more pictures of the ptosis, ophthalmoplegia in all directions, and how the same patient's leg looked with the track marks and the lesions from skin popping. So the diagnosis, the history, especially when you're getting a history of how this weakness progressed, is very important. Obviously, there's a history of drug use, things like that. And also, if the person might have eaten something, but you might not always know that you ate something suspect. But certainly if other cases have been diagnosed, and once you keep seeing patients come with the same thing, it's a lot easier. A screening tool, which is quite helpful, but not that specific, would be to look at a patient who has no fever, has the symptoms of the diplopia, the change in voice, dysarthria, dysphagia, so the bulbous symptoms and the eye symptoms, and the signs of the fixed pupils with the descending paralysis. And then you should be thinking of that. There is a differential, obviously, and I'm sure these are things I would probably always think of before I ever thought of botulism. So myasthenia gravis and Lambert-Eaton, but these are much less fulminant, and they don't have autonomic involvement. They I think, a bit more insidious. Tick paralysis, did not know about this, but as long as that tick is attached, there is paralysis. So you'd have to do a careful examination, thorough examination. You would find the tick on the body still. And then the classic Guillain-Barré syndrome, which probably is one of the ones in peds we think of first when we see a child with weakness, lower motor neuron signs. They often have some sensory symptoms. Usually it's sort of a deep muscle pain, but they do have some pain. And when we see them already with their weakness, they've already lost their reflexes. And that happens earlier than it does in botulism. And it hardly ever starts with the cranial nerve dysfunction. And they should have normal pupil responses. There is the Miller-Fisher variant of Guillain-Barré, which has the ophthalmoplegia and the other cranial neuropathies, but it's associated with prominent ataxia, which you shouldn't see in botulism. Polio is something we always have to consider, but I say apparently because I haven't seen any cases of polio, as far as I know. They should be febrile on presentation, and typically they don't have this symmetrical weakness. Magnesium intoxication can do it, diphtheria can do it, certainly organophosphate poisoning we see, and they can come in with either cholinergic or anticholinergic effects. So that is something to consider. And then probably also it could be as acute, something in the brainstem, suddenly presenting a brainstem infarct or a lesion like a neuroblastoma. So we have a topic in pediatrics, the approach to the floppy infant, and that is a child a weak child who's actually floppy versus the spastic cerebral palsy. And one thing we often think of is spinal muscular atrophy. But their history would be different. They would have a much longer history of weakness with delayed milestones. It wouldn't suddenly occur. And usually the cranial nerves aren't affected, but they do have some facial weakness and a weak cry. Then pretty rare are metabolic disorders, of really rare ones. Like I said, Guillain-Barré syndrome, but usually the age is different. We usually see that in older children. And then I suppose we would often consider meningitis, particularly TB with the cranial nerve involvement, or an encephalitis. But obviously those all have specific findings that if you're thinking botulism, you should be able to exclude the one. If you're thinking any of these, you would exclude them with imaging and probably a lumbar puncture, and over a longer time, some genetic studies. But they say if you're thinking of infant botulism, it's an emergency, so you have to act fast. And we must always remember to notify acute flaccid paralysis for 1 week, considering polio, suspected polio, and in itself, botulism is also notifiable. So in terms of making the diagnosis, you need to demonstrate the toxin, and you can use gastric secretions or vomit, serum, stool, and then also look at the suspected food source. So traditionally, they did a mouse bioassay. It took me a while to understand what happens here, but there are a lot of mice are involved. So you inject the mice, a group of mice, intraperitoneally with the sample, follow them up for symptoms, and it should develop rapidly depending on the amount of toxin you're injecting. So it could take a few days. And then once you see that they're ill, now your next step comes in. That mouse will die. Now you start with some fresh mice, and you inject those mice with each with one of 7 known type-specific botulism antitoxins. And then you inject them each with a sample that's been proven to be toxic already. And now you watch the mice, and the ones which are protected by your specific antitoxin allows you to identify which toxin type that sample contained. It allows for confirmation and toxin typing, and you can achieve that in about 75% of cases. It's labor and resource intensive, and some might say inhumane. It's not performed in all laboratories. And it also, the big thing is it also takes quite a few days. I think each step takes about 2 days. So you don't get an answer quickly. So mass spectroscopy is a novel method that is more sensitive and much quicker. And that's this MALDI-TOF machine. I have no idea how it works, but I know, having been in the lab, that they talk about it a lot for identifying pretty much any organism you need to identify. And obviously also it can be used to identify various toxins, among other things. You can do an ELISA. It recognizes antigenic binding sites. And you can, again, perform this on clinical specimens and the food samples. It is less sensitive than the mouse bioassay, so it's used as a screening test. You can also do PCR and for detection in stool and food, and that's specifically for toxins A, B, E, and F. And one of the other investigations could be electromyography, but there will be specific findings on it with repeated impulses and increments. But apparently it's quite uncomfortable, and it's not going to give you a specific, it's not that specific, and it's not diagnostic. It might just help, and it could help differentiate from another neuromuscular condition. In the laboratory, you can culture the organism from stool, serum, and also the food. You need anaerobic cultures, and it's quite difficult to grow because it's a strict anaerobe, and you need very strict conditions. There's also competing fecal microorganisms. The problem is isolating just the organism from food in the absence of proving toxin production means it's not, you can't be sure that this was the cause of the patient's illness because it is a ubiquitous organism. So you need to find that it was producing toxin. Stool cultures may remain positive up to a month after presentation. So this is on a blood agar plate. This is a metronidazole disc, so it's sensitive. So there's a zone of clearance around the metronidazole disc. And that's a gentamicin disc apparently, and it's resistant to that. It sort of makes translucent or gray colonies. They can be small and have smooth edges or irregular edges with a little bit of hemolysis. And then this is a picture of our gram-positive stain with the endospore, subterminal endospore. So the management is mostly supportive. By offering patients ventilation, mechanical ventilation, it's had a huge impact on mortality rates. And it would be indicated if they have upper airway incompetency or poor vital capacity. There is a heptavalent equine-derived antitoxin which you can use. And it's an antibody to toxins A to G. It's used for non-infant botulism. So it comes from a horse. And it's the best effect if it's given within 2 days of symptom onset. There is a 1 to 2% chance of anaphylaxis, apparently. So for infants, they use this immunoglobulin called baby BIG. So it's botulinum immune globulin. And it's a human immune globulin because they say they don't, I think my understanding was they don't want to expose the baby to horse antigens and, I don't know, predispose him to reactions later in life. So they want a human immune globulin, and they're also worried about that risk of anaphylaxis in the babies. It can really decrease the need for ventilation and time on ventilation and hospital stay. It's again, the earlier you treat, the better. And so you should treat within 3 days of hospitalization. What I don't know is whether we have access to this in our country. Antibiotics are not that helpful. So penicillin, Pen-G or metronidazole are often used, though it's untested. So it's not advised in infant botulism because then you actually could cause things to get worse by lysing the organisms and releasing the toxin. They don't advise aminoglycosides anyway because it can also impair neuron calcium entry and worsen the paralysis, as well as the fact that they could be resistant. And there's no role, obviously, in the food-borne type because you're ingesting the toxin, not the organism. So antibiotics might have a role to play in wound botulism. Debridement is very important, and then sending the tissue off for culture. So even if a wound seems to be healing well, apparently they should still debride it and remove any tissue that could contain the organisms. Local antitoxin is of questionable benefit. They do talk about pyridostigmine, which prevents breakdown. So it increases your concentration of acetylcholine at the neuromuscular junction. So makes more neurotransmitter available for impulse, for neurotransmitter, for muscle contraction. But it's not always used. But certainly if you've got a case, I think when you gave the other treatment late, or it was too late to get the treatment, the immunoglobulin, and also if you don't have it available, this might be an option while you're supporting the patient. So recovery, supportive care is the most important. You need to bind the toxin that's in the blood and in the synaptic cleft as soon as possible. And then you just have to wait for the effects of the toxin to wear off and wait for recovery of your nerve endings. Most recovery occurs, the most rapid recovery occurs within 3 months, but there's still improvements in strength and endurance up to 1 year. But with prompt diagnosis and supportive care, the mortality can be less than 8%, and infant botulism mortality can be less than 1%. Prevention is very important, and it really comes down to, well, aside from avoiding using black tar heroin and intravenous drug use, it's about what we eat. So there needs to be correct, safe food handling and preparation. So in the commercial food industry, they use they have to adhere to strict FDA guidelines. Because heat pasteurization, including vacuum packed pasteurized products and hot smoke products, they might not be enough to kill all the spores. But basically, they've acknowledged, I think, that spores might always be present, but you need to prevent creating an environment where there's bacterial growth in the containers where there's toxin production. So even if there's spores around, they mustn't be allowed to germinate and produce their toxin. Refrigeration temperatures combined with salt content and or acidic conditions will prevent growth of bacteria and formation of toxin. For people who home can, you need to use pressure canning for a specified time. And there are strict guidelines. They need to achieve temperatures above, they're quite clear on this, more than 121 degrees Celsius for a certain amount of time. And you also need to avoid excessive air because you need a good vacuum seal, because you don't want these containers to allow any leaks, because any air that comes in could carry Clostridia. It's important, I now realize it's very clear, no honey to children under 1 year of age. They're not quite sure why spores are present in honey. I read a study where they actually fed bees spores, Clostridium spores, and then analyzed their stool and to see if it came out. Anyway, so they're not sure if bees ingest the spores or if they just carry it on them and it's in their hives and then it gets into their honey. Spores are present in honey. It's not the toxin. It's nice and sweet, so it shouldn't allow for toxin production. And so it's also important to know it's not the only way infants acquire botulism, but it seems to be the one definite preventable factor. So considering it's just in the dust and the environment, infants are exposed, but this is one thing you can prevent. And people think, well, why don't we just give irradiated or pasteurized or sterilized honey? Pasteurization only is heating to about 60 degrees, so you won't kill the spores. So it seems safer just to say, wait till the child is 1 before they eat honey. Thereafter, it would appear that raw honey has many more benefits, but before 1, no honey. Just on the canning, heating under pressure should kill the organisms. You need to prevent leaks or entry of organisms. So, and then if you're worried, terminal boiling or intense heating will then inactivate the toxin. So even if there happens to be toxin in the food, if you heat it to above 100 degrees for a while before you eat it, then you would destroy the toxin. So there's entire sort of tutorials on the kind of dents you should worry about. So it's not only expanded bulging cans, it's also dents in cans. And certain dents mean this and certain dents mean that. But I think most people would say if the can is dented, don't buy that, don't eat it. So bulging tins and containers, don't just open it to see. Do not open them. You don't want any aerosolized toxin. Certainly if you didn't know and you open it, they say if there's active bubbling on opening, not a good sign. If there's an escape of gas on opening, not good. Any deformed dented cans suggest damage or inadequate sealing. If it smells or looks questionable, don't taste it, don't touch it. But they do unfortunately say that food that's been contaminated with the toxin may actually look and smell normal. I think this is a bigger problem. It is a significant problem because tin food is widely consumed. It's wherever it's used by, it's eaten all over. It's a cheap kind of food. It requires no refrigeration. Often it doesn't really require cooking. So a lot of people consume tinned foods, and you need to make the public aware of the dangers of using spoiled tins because people are at risk. Then as for vaccines, they've been tried previously. Apparently they tried a pentavalent formalin inactivated toxoid They've tried it on, used it in high-risk lab workers and military personnel in the USA, but it showed waning immunogenicity and also quite commonly local reactions. So they don't use that anymore, but that's a kind of situation where they got the human antitoxin so that they could make that human immune globulin for the infant botulism. But there are new strategies under investigation and development. Then just one slide on the therapeutic uses. Nowadays it's used in, they say, almost any field of medicine you name, there's a use for botulinum toxin, particularly in ones where there's increased muscle tone. We know we use it in cerebral palsy before contractures develop. You can use it in achalasia, anal fissures, blepharospasm, and numerous eye pathologies, migraines, torticollis, strabismus, bladder problems, hyperhidrosis, and then obviously for cosmetic use. Thank you.
Thanks, Judy. That's fantastic. Yeah, really, really important, I think, for us to know. And just, because a couple of the questions have come in as well to me, just to draw the distinction, which you did make, but the distinction between the sort of adult forms, which are usually food-borne and with preformed toxins. So the organisms make the toxin which you then ingest, but they make the toxin outside the body, versus infant, where it's usually the spores you ingest and then they germinate and form toxins in the body. I presume this is a problem of infants, not adults, because of colonization issues. In other words, it's much easier to colonize the infant colon with a new species than an adult one. I don't know, is that the reason, or is there something else you've come across in your reading? Judy, you muted.
Yeah, but I stopped hearing.
Oh, sorry, you broke up there a bit. What did you say, Judy? You may have lost Judy. Can you hear us, Judy? Okay, we'll just wait for Judy to come back. Can I ask you, John, if you're able to unmute? I don't know if you are John Friend for the NICD. Is that the reason? Is it the issue of just the extreme unlikelihood of this Clostridium botulinum spores sort of establishing a foothold in an older child and adult's colon?
Yes, I think Judy covered that. Only if there's some form of abnormality in terms of inability to produce acid, or there's a dead leg in the intestine, but otherwise not. Yeah,
okay. And then the question here about, does microwaving destroy the toxin? I mean, I guess it depends on how hot it gets and for how long. If I remember rightly, the sort of rough guide is that you should boil your canned food for at least 10 minutes. So I presume if you achieve something similar in the microwave, that should be fine. Yeah, it's the heat rather than the microwaves which would inactivate the toxin. Yeah. And then I see you've answered a question in the chat about the PCR and versus gene expression. Are you still doing the sort of the method with mice at the NICD, John, to determine? Yes. We haven't been able to do it for a while because we haven't had our animal facility has been, well, the one was closed and then the new one it's taken a while to renovate, but it's now ready. So we will be able to start animal testing again. It is said to be the most sensitive method. And it's really the reference method. But obviously it has, people are looking to replace it with other options like MALDI and very sensitive ELISAs, but it's still, I think it's still regarded as the reference method. And we don't really have access or ability to do the other tests at the moment anyway. So
we would still, in theory, do the mouse neutralization assay. It also depends on having the appropriate antitoxin. So the antitoxin that we have available, in fact, does cover the human ones, but there's a limited stock, a limited supply. It's not that easy to obtain. Just a word about the antitoxin for treatment. So what we use for testing is a different preparation from what's used for treatment. So the testing one is high potency. It's only available in very small quantities. There's no ready availability of the therapeutic antitoxin in South Africa. One has to obtain it at high expense from a very limited number of suppliers. We do have a source that is prepared to fly it to South Africa, and in fact, that's the product that was used as the one Judy mentioned, the heptavalent botulinum antitoxin. That's the one that was used to treat the floppy infant in Cape Town. She mentioned that publication. Yes, the baby product, I think, is only available in the US. It certainly wasn't. It's not easy to get hold of. I think they reserve it for their own infant botulism cases. I think it's virtually impossible to obtain elsewhere, and there's no production of antitoxin anywhere in Africa, actually.
Okay, so that's, yeah, I guess I mean, how long would it take to get it if you assumed you had the botulinum today, let's say, sorry, botulism today? Would it be sort of a week or
you
less?
can get it in, as a sort of on compassionate emergency basis within, I think it took about a week to get it, week or 10 days to get it for the baby, which incidentally was the first case identified in this country, although I'm sure it's not really the first case ever. But yes, it can be speeded up. And in the meantime, of course, other supportive measures can be used. They don't necessarily require ventilation, depending on how severe the illness is. That infant was also given pyridostigmine, I think, because the initial assessment was possible congenital myasthenia.
Yeah, okay. Judy, it sounds like you're back with us. Can you hear us?
Yes, I can. Thank you.
Oh,
in fact,
perfect.
I've asked a question as well.
I see that. Do people given toxin therapeutically develop antibodies?
It's bad time. I should have known that, but I don't. I've just thought of it.
Yes, I think they can. I think with extensive Botox use, I think there is some risk of allergy, but I'm not sure about that. One would have to check on that.
Okay, thanks.
And then you said, oh, sorry, go ahead, John.
Sorry, just to get back to Judy's point about damaged cans. Commercially produced food in cans that get damaged. Unfortunately, there is a market for those condemned and sometimes discarded products. They're sometimes retrieved and then sold cheaply to people who can't afford the full price, and that really is a risk. And in fact, that was the case with those 2 children in 2002. There was a batch of cans that had been condemned, but it was retrieved and then donated by a charity to poor people, and that's how they got that. So it really is a potential risk. And the same has happened to, the same sort of thing has been done with cans that were damaged in a supermarket fire. They found these burnt cans for sale along the side of the road. Again, potentially damaged and contaminated and a risk. Not only a botulism, but other contaminants.
Sure. And then just one last question, just was on the chat sent to me, was why does the paralysis evolve in the particular way it does? In other words, why cranial nerves first and then descending? Judy, do you have any sense? I've lost Judy. I don't know.
Oh, really?
Oh.
Can you can't hear me?
Sorry, we can hear you now, Judy. Yeah, go ahead.
Oh, I really don't know. I'm wondering if I don't know, maybe similar to myasthenia, where your eyes are most affected because that's where the highest concentration of nerve terminals and receptors are. So it manifests actually at those sites, but I really have no idea.
Yeah, I think
I don't
sorry,
know either. Yeah, sorry, I don't know.
My understanding was it was the size of the nerve. So the smallest nerves, peripheral nerves, obviously, being cranial ones, and then obviously upper limb and then lower limb. But I don't know. Now I'm starting to doubt that. If John doesn't know, suggests I've probably misapprehended that point somewhere. But it's a good way to remember it, even if it's not true. I hope it's true, but it may not be. Okay. I think we can end there. Thank you very much, Judy, because it's exactly 4 o'clock, so we can move on to the second half.